Good afternoon. This is Dr. Daniel Guerra coming to you from Authentic Biochemistry and VerEvMed Studios in the Inland Pacific Northwest. Today, as promised, I'm going to provide a lecture on T lymphocyte molecular and cellular Diaeventontology. I'll be covering a lot of ground here. I'll probably be breaking up this lecture into at least two parts, perhaps three, so that they don't get overlong each time I produce a segment. So we will get started exactly now. Okay, so again, this is my title page, so I'll just reproduce what I just said. This is VerEvMed and Authentic Biochemistry video presentation. Today is the 7th of November, 2019. Those are my two logos for the companies that I own and represent. There is my email address in case you want to contact me directly. I usually respond within a day or two. Sometimes this time of year it gets a little bit more hectic. It might take a little bit longer than that. But however, you can look at my Facebook page, all of my lectures, and my Authentic Biochemistry podcast are all being posted on that Facebook, so you can get them there. Eventually, I'm going to create a different domain and put everything in a new domain, but right now we're still using Facebook. Those are two places also you can go, these two links here. One is where the podcast is, and the other is where all these lectures are on my YouTube channel. Okay, so let's get started with this. First of all, I want to introduce, and for some of you, reintroduce a concept that I've generated called Diaeventontology. Diaeventontology describes what I've learned over the last 35 years as a PhD biochemist, physiologist, and molecular biologist. And I've condensed my understanding of those disciplines into a new conceptus, a new way of examining scientific interactions in living systems, particularly the human existing individual. But this can probably be adapted to all other systems that have as an acuity a scientific perspective. So what is Diaeventontology? What do I mean by that? You're not going to find a dictionary because I coined the term, so I have to pretty much explain it, right? It's an adaptational, processive accounting of the rational human mind through cellular and molecular event ontology and original conception. So I want to emphasize it's about the human mind because the existing individual is using the central nervous system to notice, probe, acquire, and assent to its environment. And that interaction, the human interaction, is very specific. And indeed, all of human health and all of human disease, as we know from biomedical literature, is not to be grouped together, but in fact is individual. Individualized medicine is what's being called now since almost about the beginning of the 21st century. But each human being, because we interact in the environment discreetly and uniquely, have adaptive processes, which are ultimately controlled by the central nervous system, that change our physiology, our metabolic pathways, and indeed our epigenetic immuno and neuroimmunological processes in such a way that it defines us as an individual. So everything that we've encountered in terms of pathogens or non-pathogenic microorganisms, our nutrition, our daily patterns of living, including exercise, of course, our aging process, our learning process, all of this is encountered by the central nervous system through what I call the neuroimmunoepigenome. Okay. As we go through this particular lecture and we talk more about the diaeventology of T lymphocytes, I hope that we'll be able to reveal more precisely what I mean by that process. But right now, understand it's a dialectical system that doesn't look at substance ontology, but rather event ontology. So it's dynamic. And that event ontology then helps describe the living system as it moves through the environment. So it's adaptive, as I said, and it's processive. So you have an immunoepigenome, and there's an induction of neural, endocrine, and metabolic response to the micro and macro environment. Let me explain that for a moment. The microenvironment is internal, right? All the cells in the entire system in your body. That's what I call the microenvironment. So individual cells, individual nuclei, mitochondria, and plasma reticulum, Golgi apparatus, membrane potentials for individual cells, and then cells collected in masses, and masses collected into tissues, tissues into organs, organs into the entire homeostatic human body. All of that is what I mean by the microenvironment. The macroenvironment, of course, is everything outside of us, right? And that includes our nutrition. That includes stress. That includes being impacted by potential pathogens, right? So you get the idea. Also light, heat stress, and the overall vagaries of existing in the environment as an individual. So that micro and macro environmental through classical constitutive surveillance and acquired effector cellular humoral defense stratagems, that's the entire immune system, using a reversible covalent modification. Now, this is going to involve the epigenetic processing, and of course, the hydrophobic interactions of nucleic acids, proteins, and in particular, lipids, not just lipids in membranes, but also signaling lipids. This probably, I should say, will become Diaeventontology, the central molecular theory for a free will-driven, agency-based, existing individual adaptation, and indeed knowledge-acquiring epistemology that explains the core event ontology of human existence. So I'm saying that this is a way of examining, a processive way of understanding how the individual, the existing individual, can be acquired and can be understood and can be treated in terms of health and in terms of longevity and in terms of reproductive capacity as well, of course. So that's what we're getting at. So there's the diaventome, and this is the way I kind of look at it as a trigonal planar system. You have the genome on the upper left, you have the environment on the upper right, and then you have the immunoepigenome down at the bottom. So what do I say this is? I say this is biological networks, and they are processes, and they become eventual rather than substantial in nature as opposed to substantial, okay? An event ontology that reaches across the biological network of genetic and environmental interaction is articulated and synthesized through epigenetic modification, that is chromatin alteration without having specific discrete signatures on sequence, DNA sequence, right? Epigenetic modifications of the classical and newly emerging immune system. When I say newly emerging, we're learning more and more about the immune system all the time, and the fact that it's not simply a process to ward off pathogens. It probably has a lot to do, very likely has a lot to do with the entire interaction of the existing individual in the environment. That includes learning processes, okay? Written and large, if you think about that, that's enough to keep you busy probably for the rest of your careers. And I say that it is responsive both plastically and elastically. And by that I mean the traditional rheological definitions of plastic and elastic. That is, a plastic interaction in material systems has to do with two things or more than two things interacting, and then after they interact, there's an impression left behind. And that impression that's left behind goes forward vectorially to alter all the other subsequent downstream processes. That's a plastic interaction in terms of material rheology. Then there's the elastic, which means two or more systems interact. They interact and they change, but that change goes back to the way it was before, or seemingly goes back to the way it was before, probably more stochastically or pseudo-stochastically, pseudo-randomly, than actually becoming truly elastic. But I want to get both of those concepts in there, plastic and elastic, plasticity and elasticity, in the interactions of multiple systems, including within the diaventome, the genome, the environment, and the immunoepigenome, for example. All right, so that's the focus, right? And you get a metabolic scanning, focus, accession. So first of all, there's a scanning of the environment, both micro and macro. Then there is a focus, or attention is generated. This is like stoicism for neurobiochemistry. Once the focus occurs or attention occurs, then you get an accession, meaning that there is an acknowledgement, that is, there's a reception of some new information that is processed sufficiently to be acquired or apprehended. Then there's mediation, that is, that there's a metabolic or neuroimmunoepigenetic consideration of the new information, right? It's a molecular level, the cellular level, and the whole organism level, as ordered by the central nervous system and the self, right? So that's what I mean by mediation. Then there's a learning, which means an encoding process. This, of course, has been well described in the central nervous system. And then finally, reacting to that stimuli in some way that alters behavior and alters probably the trajectory of what goes forward. So once you learn something and you learn it well, you carry that with you for the rest of your life until your memory starts to fade, or if you stop thinking about what it is you've learned. Both of those processes, unfortunately, can go into the pile of leaves that can occur in autumn. And if you bury your ability to remember with that extensive amount of dry leaves, what happens is that you lose or forfeit what you once knew. And so what you need to do is constantly recollect that, taking a Platonic term there, and recollection then involves reacquiring memories from, again, certain nuclei in the central nervous system, such that that recollection itself is a process, an eventual process, which generates an ontological perspective that allows the organism to continue to move forward with the knowledge it has acquired over space-time. Okay. So the pattern of events appear as energetic and material dynamics, much like chemical bonds. That's another very important thing. Understand that there is basically a Gibbs free energy process going on. There's enthalpies of reaction. There's entropy of decay, and there's an overall free energy, which is, when it's maintained, negative, which is constantly in the acquisition phase. So as long as the organism is alive, it's in a dynamic disequilibrium where it has a negative delta G, a negative delta Gibbs free energy. And if you actually reach equipoise, or you reach pure equilibrium, the organism stops existing, right? Dies. So living systems are always in a dynamic disequilibrium, right? That's what I mean by that. And all those chemical bonds, covalent bonds, ionic bonds, hydrophobic interactions, salt bridges, all these sorts of things we talk about in chemistry, are sort of the furniture that's involved in all the molecular interactions that are going on with the entire body, as organized by the central nervous system. So another way of looking at this in a more composite way is the existing individual, okay, and all the things that make up the existing individual, all those molecular events, because remember, I'm a biochemist, all of that existing individual agentically, that is purposely causing things in its environment and interacting through the events obtained across the inherited genome via gene expression, the changing environment, which can be random or pseudo-random, pseudo-stochastic, and the receptive architectonic. That means it's a receptive system, yet it has a structure and order. It has an architectonic neuroimmunoepigenome through time. So this is something that occurs with a delta T in the denominator, changes through time. All right. So what kind of things are we talking about? What are die-events in physiology and pathophysiology, for example, things that biomedicine is always interested in? There are molecular and cellular events that occur across mechanisms that result in real-time modifications to changing conditions. Mechanisms include, but they're not limiting to, replication, recombination, and repair of DNA, transcription, alternate splicing, and translation of RNA to polypeptide, signal transduction phenomenon across membranes and within cells and across entire systems, right? Through the humoral and through the lymphatic systems, for example. Membrane fluid dynamics, which is separate and distinct from all of this, because membrane fluid dynamics can happen at a level much quicker than sometimes even pure chemical processes, more at the level of biophysical phenomenon. You also have a cell fate repertoire, which includes things like autophagy, apoptosis, programmed cell death, that is, senescence, and necrosis. Those are all cell fate mechanisms, right? Which I've talked about at great length in my lectures here. There are also developmental programs that are superimposed upon this. This has to do with the genomic makeup, right? And that includes things like embryogenesis and alteration and differentiation, right, of different tissues and repair of those tissues, such as after wounding. And then there is, written probably largest, cellular division and differentiation, including processes like adipogenesis, neurogenesis, myelocytogenesis. Let me move my box here. Lymphocytogenesis, something we're going to talk about a great deal in these next few lectures. Gametogenesis, during the reproductive phase of the organism. And oncogenesis, and anything else you might think about to put under those major headings of how there are processes that occur in living systems. Okay. All right. So what I want to do whenever I think about a biochemical problem, remember I'm a biochemist first, is I try to write a dialectic. Now, dialectic can be looked at in various ways. Hegel rewrote it in the middle 19th century, in the 1840s, 1830s, 1840s. And what Hegel tried to say was that you had a thesis, an antithesis, and then a synthesis. I'm sure most of you have heard that, right? So that's a nice way to look at a dialectic, and it allows you to resolve contrarian phenomenon. But the original dialectic that is written in Platonic dialogues is, I think, more foundational, and I think is a better way to look at how, at least I'm trying to understand, biological phenomenon. So let's take a look at, let's create a dialectic for the specific topic. And upon doing so, that will reveal what I mean by dialectic. So the dialectic is going to come up with a statement, something that we see in nature, that we believe is accurate about nature. And then we're going to come up with not theses, but hypotheses, which means below the level of a thesis, to be able to support or deny the major concept that we are trying to examine or analyze. And that's what the dialectic is about in Platonic dialogue, in Platonic philosophy. So let's now put it into T lymphocytes, right? T lymphocytes react in the intact human dynamic event ontology to respond to the environment, maintain cellular and physiological health, and to prepare for future change. That includes nutritional alteration, neurological imprinting, disease, and indeed overarching aging. We all die, right? And before we die, we age. Senesce. So that's the major concept we want to examine. What are some of the things we need to consider in order to examine it? What are some of the concepts, conceptual phrases, or hypotheses? First of all, T lymphocyte lineages and associated biochemical communication among them are modified via changes in the epigenome, which of course I will describe in great detail as we move on, as well as the canonical inducible and repressible gene expression that we know occurs, and membrane recombination, that is retailoring of the membrane. Very important. I'm a lipid biochemist, so I would never leave that out. I would never ignore membrane recombination. That is a retailoring of membrane fluid dynamics at the first level of understanding. Of course, all the chemical processes therein. So that's one thing to consider. The second thing to consider is there are key epigenetic molecular events in that dynamic process. And they include, specifically, chromatin remodeling itself, transcription factors, alteration of chromatin because of protein DNA interactions, lipid protein DNA interactions, these kind of chemical and biochemical and biophysical phenomenon. CpG methylation, that is when you get a specific carbon 5 on a cytosine residue, becomes methylated by a DNA methyltransferase on the DNA sequence bound up in the chromatin within the nucleus of the eukaryotic organism. Histone acetylation, remember the histones are part of the nucleosomal complex, right? You have an octet of histones, these basic proteins that wind DNA up and put it inside the nucleus, package it. So you have histones, specifically lysine residues, which are acetylated. Substrate for that, by the way, is acetyl-CoA. Substrate for methylation, by the way, is acetylmethionine. And their metabolic interaction in the nucleus, so that you get this epigenetic reprogramming, are also part of how bioenergetics plays a role in the whole process. All right. You also have microRNA, which I've talked about just recently in a lecture. And microRNA are basically these often interfering RNA, so microRNA interference. And what they interfere with is the translation of a messenger RNA. Okay. Now, microRNA can also work at the level of transcription, controlling transcription by making DNA-RNA hybrids in erstwhile heterochromatin complexes. So all of those processes I just described to you, chromatin remodeling, CpG methylation, histone acetylation, that doesn't include everything, by the way, and microRNA interference, everything I'm meaning in what I call epigenetics. But those inclusive require the establishment of diaeventological phenomena. Okay? And that means the process I just talked about, right, where the environment, the genome, and the immunoepigenome are interacting in the simplest way, in a trigonal planar way, to generate all then extending processes subsequent to that induction phenomenon and acquisition phenomenon, apprehension acquisition phenomenon. All right. Now, we're going to look at a paper that was published about a year ago. It was actually published in December, late December of 2018, in one of my favorite journals, Journal of Biological Chemistry, and there's a citation. Okay, so what does this paper talk about? Now we're going to get very specific, because again, this is biochemists talking to you. So this is authentic biochemistry, right? And this is also an evidence-based biomedical consulting service, which is what we run. And so what I want you to understand is that we look at specific published research articles. What I just described to you for the last 15 minutes or so is my approach at examining that published peer-reviewed scientific literature. Now, I want to emphasize, I don't look at, read, or respond to any popular press articles. I don't really respond to, at least in this format, books, which could be well referenced, right? Well footnoted, as they say, but nevertheless exist as a frozen structure that's already in the past by the time the book gets published. So I like to look at brand new events in the scientific literature, or when I go back to cohere all of the previous literature, like this late 2018 paper, to what's being published now. You can't just look at current published papers, because you need to know the foundation and coherence of where they come from to be able to come up with molecular and biological truths, right? And the other way you can really assume that is by understanding and apprehending all the previous literature that's necessary to acquire that knowledge. Okay, so what's this paper about? Since methylation can be an off-on switch for chromatin remodeling mediated nascent transcription, both the combined accumulation of covalent histone methylation, histones can also be methylated and acetylated, as I previously mentioned, and the sequence specificity must be read out according to transcript level, like individual discrete transcripts, right? Specific genes being transcribed. In particular, the methylation of a histone H3 at lysine residues 4, 36, and 79, shown there, is associated with transcriptional activation, whereas methylation histone H3 at lysines 9 and 27 is correlated with transcriptional silencing. So if you know something about epigenetic programming, you might think that, well, methylation reads silencing, removal of the methylation, demethylation reads activation or expression. Not the case. There's a very specific histone code. Certain methylation patterns actually allow enhanced expression of genes. Other methylation patterns suppress gene expression, even at the histone level. And of course, we also have methylation, acetylation, sumoylation, ubiquitinylation, other kinds of covalent modification like succinylation of those cytosine residues in CpG islands, and even get phosphorylation patterning, certainly on the protein level with histones and other transcription factors and single-stranded binding proteins and such that are involved in transcription translational machinery, opening up chromatin remodeling and making the transcriptional bubble, and ultimately translation, all the splicing and processing of the nascent RNA even before it leaves the nucleus, and certainly subsequent to it, and all the ribosomal RNA machinery as well, that ultimately end up making translatable product. And then all the targeting those proteins that are specific intracellular and intracellular subcellular regions and outside the cell via glycosylation patterns that are associated with the endoplasmic reticulum, Golgi apparatus, and then moving through endosomal processes through and inside and out the plasma membrane. All right. So more about the specific things in this paper. Histone methylation and demethylation are complex elastic phenomena. I told you about elastic. They occur, they have an effect, and then they are removed. Okay, they are removed, or at least there is a pseudo-stochastic realization at the molecular level that they've been removed. We don't really know if they don't leave a molecular footprint. It's a totally different story, right? All right, but the phenomena itself are elastic. In other words, you can methylate DNA and you can demethylate it. And so quickly, unless you have the right experimental design, basically you're looking at how methylation patterns happen inside specific pieces of DNA that you've isolated, and you've used proper probes so that you can determine what cytosine residues have been methylated. And with the histones, it's even more complex because you have to isolate the histones in real time. Because there's methylation and demethylation, you have to catch this, and that's why substance ontology doesn't work. It has to be event ontology. You have to follow the process. So any one isolated event doesn't tell you anything. One isolated event is actually basically just a substantial phenomenological system. What you want is to have it over time and over space, too, depending on what cells you're looking at. So methylation and demethylation are elastic phenomena. The methyltransferase called EZH2, and that stands for, I know you like this, enhancer of zesty homolog 2, probably from Drosophila molecular genetics. Thank you, Dr. Morgan. Catalyzes the S-adenosylmethionine-dependent trimethylation of this histone 3 lysine 27, right, those two. Members of the Jumanji domain containing, that's JMJ, Fe2 and Fe and 2-oxoglutarate dependent oxygenases. Okay, so that's ferrous iron and 2-oxoglutarate dependent oxygenases, two different oxygenases working upon the redox potential of those two compounds, either ferrous iron directly or in chelated form, and then because of 2-oxoglutarate. Those oxygenases are catalyzing the demethylation of methylated histone lysines. Now, why am I telling you about the specificity of the iron and 2-oxoglutarate? Because you need to understand them. You have to go back one level and say, how do you end up with iron in that redox state? How much of the iron is available? What else is it interacting with? Transferrin, for example. Is it bound in hemoglobin? Is hemoglobin breaking down? Right? Or is it being transferred in the mitochondria? Or is the iron in the process of being reduced or oxidized? Right? All of these are necessary molecular events which will lead directly to the demethylation process. And those also have to be included in the understanding of the dialectical event ontological perspective that's going on in the cell in real time. Right. Members of that, okay, as I just said this, sorry. So the JMJ-D3 and the KDM6B and the ubiquitously transcribed tetratricopeptide repeat gene, X chromosome UTX and KDM6A are demethylases. Those are all demethylases of that histone 3 lysine 27 ME23. And the UTX appears to be constitutively expressed in many tissues. So are these demethylases expressed all the time or not? Some are, some aren't. See how this works, right? So there's on top of it, just a genomic expression of these genes at the level of protein, transcription translation of the protein. Proteins ending up back in the nucleus have to be targeted back into it. right, through the nuclear envelope. All of that is probably a very non-random process. But you have to know if those genes are being expressed via induction or are they housekeeping genes? And therefore the proteins are there all the time. You have to ask those questions too. Again, back one level all the time. All right. The JMJD3 is inducible by, look at this, lipopolysaccharide, foreign antigen from bacteria. Several cytokines, mostly pro-inflammatory cytokines, it's glycoproteins generated from things like T lymphocytes and growth factors. Okay, growth factors, of course, also control gene expression. Both those enzymes play important roles in macrophage activity, switching from M1 to M2, which is the quiescent macrophage, development of the entire system, all cellular nuclei and masses, and stem cell function, right? The generation of new vascular beds, for example, and things like embryogenesis, right? And osteoclastogenesis, things like this, processes like that. Adipogenesis that I was talking about at the very beginning. All right. UTX is a component of a transcriptional complex. It's a demethylation part of a transcriptional process, such as trithorax and MLL, which is actually also known as mixed lineage leukemia, which actually regulate gene activation. They're named after that. Regulate gene activation through the histone 3 lysine K4 moiety, and that methyltransferase, and then the H3K27 demethylase activity. So these enzymes are interacting with one another at the protein-protein level, at the protein-DNA level, and at the protein-RNA level, interacting in very specific ways and getting into the nucleus and altering the methylome via demethylation. That's what this paper was about. Okay. So I've got way down into it right now in the early stage of this lecture. Not so early anymore, by the way. Why am I doing that? So that you get the full understanding of where we're going to go. Okay? It's fine to be at the 100,000 foot level above the surface of the planet and talk about the Diaeventology of this whole system. But we have to get all the way down to the molecular, even the submolecular level, the atomic level, to fully understand the process as it occurs authentically in real time. Okay. All right. There's a paper published in Cell, Death, and Disease, which I've looked at recently, published in just 2019, not very long ago, actually, and there's the citation. Okay. I mentioned this before, and I'm repeating it now so that we can talk about this Diaeventontological perspective of T lymphocytes. Inflammatory bowel disease, including Crohn's and ulcerative colitis, CDUC, associate with an intestinal inflammatory lesion where group 3 innate lymphoid cells called ILC3s, now we're at lymphoid cells, okay, lymphocytes, physiological relevant and healthy GI mucosal homeostasis through moderate generation of the following cytokines, IL-22, 17, and the GM-CSF, okay, so that's a growth factor. All of those become dysfunctional when you get IBD. And it's coming from the expression of these cytokines and growth factor there are coming from these unique group 3 innate lymphoid cells, a specific type of lymphoid cell, right? So ILC3 can contribute to the progression and the aggravation of inflammatory bowel disease, while both IL-22 and IL-17, those are the cytokines, along with interferon gamma, are overexpressed, and they are overexpressed because of the dysregulation of the following: NCR ILC3, NCR negative ILC3, or NCR positive ILC3 function. Now, what is NCR? You can either have NCR being expressed, it's a receptor, it's called the natural cytotoxicity receptor, NCR. It's a type of a type 1 transmembrane protein of the immunoglobulin superfamily. That's correct. And upon stimulation, it mediates another lymphocyte, natural killing and release of interferon gamma, usually a pro-inflammatory cytokine, and also growth factor in some cells. They bind viral ligands, typically, such as hemagglutinins and hemagglutinin neuraminases, some bacterial ligands, and cellular ligands related with tumor growth. And sometimes when you get autoimmune diseases, they make a mistake and natural killer cells go after naturally occurring host antigen complexes on cell surfaces. Okay? So there is a progression towards the NCR plus, the natural cytotoxicity receptor, which mediates this phenomenon I'm just telling you about through this receptor-mediated system, okay, on the immunoglobulin type receptor. There's a progression towards the NCR positive, means NCR is being expressed, that's what positive means, ILC3 type cells, and then transformation of that NCR positive ILC3 to a new cell lineage, ILC1, as well as regulatory ILC dysfunction. That means no longer regulated by all these cytokines, chemokines, et cetera, growth factors, into a pathological state. Now you're starting to get into what's going on with IBD. So I want you to think about what I told you just before about this entire phenomenon, about how intracellular phenomena associate with all of these growth factors and cytokines and chemokines. Now I'm focusing in on the fact that they interact in a particular disease. The larger common understanding of disease is inflammatory bowel disorder, but also specifically UC and Crohn's disease. Okay. You're getting the point here. Now, let's summarize what we have so far. Group 3, innate lymphoid cells, ILC3s, are responsible for GI mucosal homeostasis. So they're supposed to be expressed, they're supposed to be found there, they're supposed to be attended there and apprehended in that cell system, in the mucosal system of the GI tract. Okay? And they exist there, and they moderate the equipoise of the immune response in the gut, which is basically exposed directly to the external environment because of diet, right, for example. Because they have moderate generation, as expression of, I just said, interleukin 22, 17, and the GM-CSF. It's growth factor, right? Colony stimulating factor. Okay, now when that happens in a pathophysiological state, there's a dysregulation, that's when you get the disease. Next statement. ILC3 contribute to the progression and aggravation then of inflammatory bowel disease by the dysregulation of the NCR, remember that receptor, minus ILC3s. So even when the receptor isn't expressed, or certainly when the receptor is expressed in the ILC3 lineage. And the bias of the NCR plus ILC3 lineage will then lead to the proximal differentiation, because of changes in gene expression, to ILC1 lineages, right? These innate lymphoid cell lineages, under the stimulation of a new cytokine, interleukin 12. That's generated by a different cell lineage that's been recruited, the CD14 positive dendritic cells, which you know are antigen presenting cells, as well as a regulatory ILC dysfunction in the pathological state, which we've been describing, we'll get into great detail about. So the dysregulation of ILC3 results in the overexpression now, not the low-level expression, but the overexpression now of these actual pro-inflammatory cytokines, interleukin 22, 17, interferon gamma, in which IL-17 can recruit this new cytokine being generated at high levels, so it reaches the correct concentration. It starts to recruit now innate immune cells of the neutrophil class, and they disrupt E-cadherin. Okay, it's a cohesion molecule, and junctional adhesion molecule-like molecule, JAML. Sorry for the name, I didn't make it up. Blame the geneticists always. Leads to the enhancement of epithelial permeability paradigmatic to inflammatory bowel disease. See how that works? So the ILC3 to ILC1 plasticity means you make the change and it does not revert, plasticity, is reversible, okay, over time in the presence of, so that is the conversion is reversible. But once you make that differentiation to ILC1, depending on what's going on in the microenvironment, that might be actually terminally differentiated, you see. So the process is reversible, but not necessarily the products of the process. Okay, and that happens in the presence of interleukin-23, interleukin-1 beta, and the retinoic acid produced by CD14 dendritic cells. Why is retinoic acid important? Because that's a lipid, right? When you hear about retinoic acid, because retinoic acid is associated with retinoic acid, the gamma ROR gamma T receptor system, which regulates all the T lymphocytes. Okay, so those are orphan receptors, right? Retinoic acid responsive orphan receptors on T cell lineages, which are turned on by retinoic acid, and then further develop and differentiate into different T cell lineages. What types? Th1, Th2, these H stands for helper, or they used to be called effector, Th17, and Treg cells, which, if they're in the correct expression patterning, will suppress the activity of Th1, Th2, and Th17, which are usually considered to be pro-inflammatory. Right? See how this works. All right. So, more from this paper about ILC3s. ILC3s express the retinoic acid receptor, RAR, related orphan receptor. Now it's called ROR gamma T. It's a specific subunit form of it. Sorry, I'm going to move myself again. And these signature cytokines, interleukin-22, 17. You also have this fetal type of innate lymphocyte, C3, or just lymphoid tissue inducer cells. Those are required for overall lymphoid organogenesis. Okay? While postnatally developing ILC3s are important for the generation of intestinal cryptopatches, that's what they're supposed to do, and isolated lymphoid follicles, as well as for the defense against pathogens and epithelial homeostasis, and therefore generating epithelial homeostasis. That comes from a journal called European Journal of Immunology, published, you can see there in 2015. So see, what I do is I go into the primary literature and I'm explaining to you in more detail how these receptors function. That's my job. So group 3 innate lymphoid cells, the ILC3s, are responsible for the GI mucosal homeostasis that we've already been saying through moderate generation of those cytokines and the stimulating factor, colony stimulating factor. ILC3 contributes to the progression and activation of inflammatory bowel disease, as I've said, through those two systems. And finally, the ILC1 and 3 plasticity is reversible. Okay. Now, here's a paper published in Frontiers of Immunology back in September of 2015. We need to know this so we can continue with our discussion. What does this paper tell us? Regulatory T cells, Tregs, via their immune suppressive capability or capacity, play an indispensable role in maintaining immune homeostasis and preventing autoimmunity induced by excessive, misdirected, misguided, or unnecessary immune activation. That is, for example, mistaking an autoantigen, a self-antigen, for a pathological antigen. For example, like LPS, or say a sialic acid residue on an epithelial cell for, say, LPS. Okay. Surface expressed cytotoxic T lymphocyte associated antigen 4 mediates the suppression of the target cells by a cell-cell contact. So you have to have cell-cell contact for that antigen 4 to work. Okay. So Treg cells can also reduce T cell activation of the helper type and proliferation, okay, that is division, through the CD39, CD73 mediated production of metabolic adenosine. While Treg cells also, that's a nucleotide, of course, while Treg cells also have been shown, adenine nucleotide, right, so it's a purine, have also been shown to harbor cytotoxic capacity and induce target cell apoptosis, programmed cell death, through release of granzymes A, B, and through perforin. Anti-inflammatory cytokines that are secreted by Treg cells can also induce immune tolerance, right? Immune tolerance, which is the healthy system usually. Under pathogenic conditions, however, such as systemic lupus erythematosus, SLE, and multiple sclerosis, two terrible human diseases, Treg cells exhibit plasticity to some extent and may mimic T helper-like phenotypes. Okay, sorry, I was down and low on through all that discussion, but there I am back again. All right, so we're going to stop there, okay, and we're going to continue on with this lecture. I'm going to give you more of that phenomenon you see there. It's going to take you to the goodbye slide where we started from. I'm going to go back and I'm going to look at T cell differentiation now in a very processive event ontological methodology so that you can understand how these T cells interact with one another in combination with cytokines and chemokines. That's really important because we're not going to be able to ever get to an understanding of how this dynamic system works relative to autoimmune diseases, and that's where I'm leading to. I'm moving on to autoimmune disorders eventually with this arc of lectures. So let's close this. Close that. And we're going to leave right now. We're going to say goodbye to my ma. Goodbye, Ma. Hope you're watching. And we'll get back to you very soon with part 2 of this hopefully very stimulating and interesting arc of lectures on the Diaeventontology of T lymphocytes. So this is Dr. Dan Guerra from VerEvMed and Authentic Biochemistry saying bye for now.